The second part of the week one lecture is all about fluorescent proteins and what I think is the bread and butter of doing cell biology research, live cell imaging. So fluorescent proteins. I asked you all in year one, if you were a smart scientist and you discovered a protein that was fluorescent and was green, what would you call it? And I didn't get any ready answers. Very seldom does anybody come up with the fact that I'm talking about green fluorescent protein, and it's because it's a funny name. Do you not find that funny? I expect proteins to have great big long scientific sounding names. So I think the people that discovered this protein had a bit of fun calling it green fluorescent protein, but I mean, it describes it perfectly. Most of you are probably familiar with GFP by now and have seen cells expressing GFP and know what it looks like. So there's Alba the bunny. Was Alba a real bunny? Indeed she was. There's a little blurb there about how Alba was made. And Alba was made a long time ago. This goes right back 2002, 2003, something like that. And she was transformed. In the embryonic stage, she was transformed so that all of the cells in her body expressed the gene for green fluorescent protein. And she glowed green when blue light was shone upon her. Now it does say, and I've always wondered about this, it says down at the end of this little blurb that comes from Wikipedia, photos by Kak, who was the artist that helped in creation of Alba, showing the entire organism, including its hair, glowing a uniform green, have had their veracity challenged. Easy enough to take a picture of a white rabbit and color it green like that with Photoshop. So I too am a little bit skeptical that these pictures of Alba, the glowing green bunny that we see, are absolutely real. I don't think the green would look that bright to the naked eye when the whole rabbit was lit up with blue light. But in any case, nowadays we have green mammals, we have green cats and green sheep and green pigs and things like that. And if you photograph them and you collect the fluorescence that's coming off them for a long period of time, you can definitely see that there is a green glow there. So here is a protein that can be made by inserting the gene into an animal, letting the animal grow up so that it expresses the gene in all of its cells, and the protein therefore is fluorescent and the animal glows green when it's lit appropriately. Let's look at the whole story behind GFP. Fluorescent proteins in general. The most widely used fluorescent proteins are derivatives of a native protein from the jellyfish Aquaria victoria. That is a picture of Aquaria victoria right there. That glow that you're seeing is not the GFP, and this is a very, very common mistake. In the early 2000s, a lot of textbooks and journals started publishing pictures like this of this jellyfish. And it was because they were fascinated, of course, by the origin of this protein. Well, the jellyfish has GFP, but the bright glow that you're seeing here is not GFP. All that is because they're underwater and the photographer is using a great big strobe light to light the jellyfish, and it's reflecting back off of the jellyfish. The GFP, if you grab that jellyfish by the top of its crown and you flipped it up like this and you looked under the rim, down in under here, you would see these little pockets of green fluorescence. And that's the GFP. Here it is here in a more magnified version. You can see that there are little pockets of cells that contain the green fluorescence. Why does this jellyfish have green fluorescence? Nobody has ever satisfactorily answered that question. It doesn't seem as if the jellyfish use it as some sort of mating display or anything like that. Some people have suggested that maybe it has something to do with sun protection because it harvests energy from the sun and transduces it into bioluminescence and biofluorescence. So we don't really know why the jellyfish has green fluorescent protein. What I will tell you is that it's been discovered in other sort of related, even distantly related sea organisms since then. Fluorescent proteins, like green fluorescent proteins, serve as energy transfer acceptors, receiving energy from calcium-activated chemiluminescent proteins that are called aquarins. So the jellyfish, and in fact the aquarin protein was first discovered in the jellyfish before people realized that the green fluorescent protein was there to accept electrons from it. Fluorescent proteins are continually produced within living cells and subject to cellular targeting, partitioning, and turnover processes, just like all of your other proteins are. Now, the neat thing about that is this distinguishes them from normal fluorochromes. Before green fluorescent protein came along, if we wanted to make a cell fluorescent, there were a limited number of things we could do. Some fluorescent stains are taken up naturally by a cell because they partition into the membrane and then they're transported into the inside of the cell. But the thing is, they don't last in there. They're turned over fairly quickly, and as the cell divides, the daughter cells lose the fluorescence. Other stains can be injected into cells, and people have used the so-called tracer technique over the years, but it's again not ideal. A lot of these stains are very toxic to the cells, and the other thing was they didn't really target specific organelles within the cell. There is one famous fluorochrome that's called mitotracker, which goes right into the mitochondria because of the different voltage across the mitochondrial membrane. So the mitochondrial membrane has a different potential, and that means that that stain specifically goes into the mitochondria. But for example, we didn't have a stain that would let us stain the endoplasmic reticulum or the Golgi apparatus. So we had seen all of these organelles in fixed section material, particularly for electron microscopy, but we couldn't really visualize them in the living cell. We couldn't watch the organelles moving around and doing their job. So along comes fluorescent protein. It's continually produced within living cells. Why? Well, because it's a protein. A protein means that it's transcribed and translated from a gene. The gene is in the genome of the organism that has the protein. So when you see organisms like Alba the bunny that express green fluorescent protein, their genome has been altered. They're transgenic animals and plants. But because the gene is in there, the gene can be transcribed and new protein can be made to replace fluorescent protein that's gotten old and been turned over and recycled within the cell. These proteins are very bright and non-toxic, and that was a big question that we wrestled with back in the early days of fluorescent protein technology. Whenever we would try and publish papers in the early 2000s, people would say, but isn't that bright fluorescent protein, isn't that damaging to the organelle, isn't that toxic to the cell? The answer, as far as we're concerned now, is that no, it's not. It's not toxic at all. It's just another protein that's made inside of the cell, not even fluorescent most of the time until a strong blue light is shone on it. So the cell just goes along with the fluorescent protein in it. The cell can go through mitosis perfectly well. I'll show you how we use it to study mitosis, in fact, and the organisms can reproduce and produce generation after generation that all contain the gene for green fluorescent protein. So that's the beauty. You transform an organism, you let it reproduce, the next generation still has the green fluorescent protein. And importantly, fluorescent protein expression and subcellular localization can be controlled using molecular biology techniques. I told you about some of those early stains. We could work and we could get them into cells, but they wouldn't really show us one specific organelle. You couldn't mark the nucleus specifically, or the endoplasmic reticulum, or the plasma membrane really specifically. Well, we can with fluorescent proteins, and that's because there's a trick that allows us to target GFP and related fluorescent proteins exactly to the organelle where we want them to be expressed. So let's talk about the discovery of GFP. It wasn't discovered in 2008. It was discovered many years before that. In 2008, the people that discovered and learned how to use GFP won the Nobel Prize in medicine, chemistry, won the Nobel Prize in chemistry. And they won the Nobel Prize in chemistry because they completely shifted the paradigm, which is science speak for they had completely changed the way that people do cell biology. Everything changed in the late 1990s and onwards. The way cell biology was done changed, and we're still using green fluorescent protein as much now as we were in the early part of this century. Almost all research that goes on looks at living cells marked with fluorescent proteins in one way or the other. This guy, Osamu Shimomura, discovered that Achoria victoria had this green fluorescent protein. I'm not actually positive he discovered it. I think there had been reports from other people earlier on that the jellyfish actually had the GFP, but Shimomura went after it. He purified the GFP from the jellyfish, and he ascertained that it was a protein. He gives talks. Saw him give a talk one time. That little pot that he's holding in his hand is pure green fluorescent protein in solution, and it's really funny because he turns out the lights and he shines a blue laser on it, and bang, it lights up the entire stage where he's standing. It's such bright stuff. So way back in 1962, he harvested GFP from jellyfish. He tells the grisly story about how him and his research students chopped up 10,000 jellyfish to get enough GFP to really work with it and to analyze the protein structure. Anyway, he didn't do anything else with it for a number of years, and that's because in these 30 intervening years between 1962 and 1992, there wasn't really very much you could do. You could rationalize, you could think about the fact that the protein was there, and if there was a protein, that meant that the cells had a messenger RNA, and ultimately that they had a gene. There was a stretch of DNA that codes for this protein, but there was no way back in the 1960s and 70s that you could go find a gene. You could use what are called map-based techniques to figure out which chromosome a gene was on or something like that, but there was no DNA sequencing really. There was no molecular biology that would let you find a gene in a genome and cut it out and clone it into bacteria to bulk it up. Well, in the early 1990s, Douglas Prasher is a PhD student. That's Douglas there. He's a PhD student, and he's got the tools now. He's been working with molecular biology. He knows how to clone. He knows how to do PCR, all of those sorts of things. He spends years going through the jellyfish DNA trying to find the gene for GFP. And he finally figures it out in the early 90s. So he's the one that first cloned the gene out of the jellyfish and got it into bacteria. And then he graduated. He got his PhD. He's Dr. Douglas Prasher, and he dropped out of science. And he dropped out of science because he didn't get funding. He didn't get a job in science to continue with. And he went off and he worked at various different menial kind of jobs. He became a truck driver at one point. Anyway, the work had sort of stagnated there. Then along comes this guy here, Martin Chalfie. And Martin Chalfie expressed the gene in bacteria. Douglas Prasher had already put it into bacteria. Chalfie chopped it out, I think, put it into his own different bacteria, shone the blue and UV excitation wavelengths on the GFP, and lo and behold, the bacteria glowed green. Eureka. I mean, why didn't Prasher do that? Chalfie did it, 1994. Well, I mean, and I call it a eureka moment, and it really was. Think about what that means. That means that a gene from a eukaryotic organism, the jellyfish, is being expressed in a bacteria. The protein has been produced by transcription translation, and the protein actually works. It's still fluorescent, just like it is in the jellyfish. The bacteria are happy and healthy, and when they divide by asexual reproduction, the daughter cells each have the gene for green fluorescent protein. This is Thule Hazelrig, and they got married. And Thule Hazelrig did something amazing as well. She was amongst the first to express the green fluorescent protein as what's called a fusion protein, and I'm going to explain to you later in the lecture why that's important. I don't know if the dog did anything related to GFP. Jury's still out. Last person in this story is this guy, Roger Chen. That's Roger Chen right there. And he's drinking some champagne, you'll notice. Roger Chen's lab learned how to mutate the wild-type GFP sequence to do all sorts of things to it, to make it brighter, to make it last longer, but most importantly, to make it shift color. If you just fool with the amino acid sequence of wild-type GFP, you can shift it towards blue or you can shift it towards red fluorescence. So Chen's lab were responsible in the early days for making yellow fluorescent protein and for making one called cyan fluorescent protein. And that's where I started with GFP. There was GFP and there was this brand new thing called YFP. And that was very exciting back in 2000, 2001, something like that. So the Nobel Prize The Nobel Prize is awarded to up to 3 people for a discovery. And I've told you about 2, 3, 4, even 5 people here, but Tule Hazelrig wasn't one of the principal people that was in on the development of GFP itself. So if we're thinking about the development of GFP itself, and you can award to 1, 2, 3 people of the 4 I've mentioned, which 3 get it? And to cut a long story short, and I'm surprised about this, the people that get it are the guy that first purified it out of the jellyfish, the guy that showed it worked in bacteria, and the guy that figured out how to mutate it and make the different colors. If I was the Nobel Committee, I would award it to the guy that discovered it, the guy that first cloned it, spent all those years doing all that work, and the guy that figured out how to mutate it and make variants. It is my opinion that Chalfie really didn't do very much in that story. Anyway, Prasher's comment when he heard about the Nobel Prize was, well, I hope when they come through Knoxville, Tennessee, which is where he was living, or was it Nashville? One of those cities in Tennessee. He said, I hope when they come through town, they stop and buy me lunch. Chalfie went further than that. He hired Prasher to be his lab manager. So at last I heard, Prasher was actually running this guy's lab at the University of Chicago. Don't know if that's true to this day, but it's a bit of good news anyway. Prasher got back into science. So wild-type GFP has excitation and emission spectra, just like what I told you about in the first part of the lecture when I was saying that all fluorochromes have a characteristic excitation and emission. Wild-type GFP has a very, very strong excitation peak in the UV region, just shy of, where's it at, 395 nanometers, just short of 400 nanometers. And it's got a secondary small excitation peak at 475 nanometers. And it emits at 509 or 510, very, very strongly in the green region. This was a problem for the pioneers in GFP research. And it was a problem because these UV lasers that we were using in the confocal systems to excite the GFP are very, very damaging to cells. So I told you the green fluorescent protein itself doesn't do any harm to the cell, but the lasers were really, really strong. Remember, I told you UV light is down at the very energetic end of the light spectrum. People made mutants, and one of the mutants that they came up with was a 1 amino acid substitution at position 65 within the protein chain of green fluorescent protein. S, the amino acid S, serine, was replaced by the amino acid T, threonine. So the S65T variant or mutation completely abolished the UV excitation peak, and it made the excitation peak at 484 nanometers much, much, much stronger. So we've got a strong blue light excitation, and we maintain the strong green light emission. I told you about Roger Chen and his lab making all of these different variants of GFP. Well, if we look, and if you look at the names of these things here, it kind of makes you hungry with plum, grape, raspberry, cherry, strawberry, tangerine, tomato, orange, banana, honeydew, and citrine, and then these ones with the E's in front of them. Well, the original GFP wild-type sequence was so-called re-engineered. And it was re-engineered to have codon usage that mammalian cells, like human cells, prefer. So people fooled with the triplet code of the gene so that mammalian cells would produce more of the protein, produce it more readily. And that worked. And that human codon optimized GFP was called eGFP, and that's it down there. And that's a very bright variant of GFP that carries the S65T mutation that I told you about before. Roger Chen's lab learned how to fool with the sequence and shifted a little bit towards the blue, and we came up with E. I think the E stood for enhanced, if I remember correctly, enhanced CFP. And they also made one that's not shown in this list called enhanced YFP, and it was a little bit yellower. It was like this one that's called citrine. So there was a little wee bit of variation in the colors that were emitted. That was a couple of decades ago. Nowadays, we've got lots and lots of different colors. We've got fluorescent proteins that go all the way through the yellows into the oranges, and then a whole suite of ones that go through the reds, and in fact, right out into the infrared. You can't even see these ones by eye when they're emitting. You have to use a detector that detects long wavelength light in the infrared region up above 700, and you can take a picture of it that way. And there's even a shorter wavelength one that's called blue fluorescent protein now. And by the way, this picture here, although it's amazing, is nowhere near exhaustive. There are hundreds and hundreds of variants of the fluorescent proteins. Newer versions have at least a 20-fold brighter fluorescence and often produce much more. That's because of the enhanced thing I was talking about, much more than the wild-type protein. This designation here, XFP, just means whatever fluorescent protein. So you can have CFP, BFP, YFP, GFP, RFP, if you're talking about the red ones up here. They emit different colors and can be used simultaneously to monitor independent cellular events, and that's what's really cool. You can mark one cellular structure, say the endoplasmic reticulum, in green, and you can mark another cellular structure like the mitochondria or whatever in red, and you can see them both at the same time. You can even use 3 colors in a cell. I'll show you an example of that in a minute. Some GFP mutants exhibit more rapid formation of the fluorophore. So from sort of 20 hours it takes the protein to fold and mature and become fluorescent, they do it really, really quickly, and that can be important in some experimental situations. Or they work in different cellular environments, like in lower pH compartments where the wild-type GFP doesn't. One thing I didn't mention when I was talking about all of these colors here, all of the ones down at this end were developed from the Aquaria victoria jellyfish gene. The red ones up here come from reef corals. So they come from organisms that are related to jellyfish. Once people found out that the jellyfish had the green fluorescent protein, the hunt was on. People went scuba diving and they started collecting different animals on coral reefs to see if anybody else had fluorescence. And of course they came up with a bunch of corals that had a protein that was much, much redder than the green one that came from the jellyfish. And this picture here is a molecular biology vector diagram, and this is the way that we would go about getting GFP. If we wanted some GFP for our research, we would go online, go to the company, which was called Clone Tech at the time, that sold the GFP vectors, and we would say, send me the GFP vector. You would get this vector here, which you could transform into some bacteria, and it carries, there it is right there, the gene for green fluorescent protein. You could grow that in your lab. Once you'd paid for the vector, you could grow it in your lab and have a ready supply of it whenever you needed it. All you needed to do if you wanted to transform it into a different animal was clone it out of there and use some transformation technique to get it into the animal genome or the plant genome, which was my kind of research. And by the way academic research works, if you're doing academic research and somebody publishes a paper in some journal, Science or Cell or something like that, and in their paper they're talking about a new vector they've made that contains GFP and other things that are interesting to you, you don't have to pay them for it. You can send them an email and say, please send me some of the vectors. So anybody that does research in molecular biology makes their own vectors. When they get a request from somebody, I've got a few in my inbox at the moment, we're supposed to package up a bit of the DNA and send it off to them. So that's one really nice thing about academia. We share all of the intellectual protocol in that sense. But if you're a commercial organization, you still have to pay the license to use GFP. It was patented technology. Look at that. There's a classic piece of artwork. See what that is? Lovely sunset scene. There's the sun's in red and the leaves on the palm tree are in green and the sea is in a bluey cyan kind of color. We've even got some lovely purples up here in the sunset. That's bacteria on a petri dish. And the way the person did that, It's absolutely amazing. It is Nathan Shaner that did this, and he's pretty famous in the fluorescent protein world. I guess what he did was he drew the picture on a piece of paper and put it down under the petri dish, and then he had bacteria, little tubes of bacteria. This is the bacteria liquid suspension culture that contains the green fluorescent protein. And you would take a little toothpick or something with a bit of that, and you would paint where you wanted the leaves to be on the tree. Then you would go to the tube that had the bacteria with the red fluorescent protein, and you would paint over where you wanted the sun. You do all that with all the different colors that you've planned. You put the petri dish into the incubator for, it looks like about 2 or 3 days, and then you photograph it using different excitation light sources to record the different colors. And finally, you put all the photographs together in layers using Photoshop so that you produce a beautiful, beautiful scene. But nothing better illustrates the color, the variety of colors that are possible with the different fluorescent proteins than that picture right there. I think it's really cool. So what can we do with fluorescent proteins? Well, we can transform organisms. And when I first started giving talks like this 20 years ago, there were a few organisms that had been transformed. Jellyfish, I told you they were first, the gene from jellyfish was first used to transform bacteria. Okay, so we can do prokaryotic cells. But then people started with the simpler eukaryotic organisms like C. elegans, the nematode worm, or Dictyostelium, the slime mold. And of course you can transform them relatively straightforwardly, and you can make them look bright green. Well, over the years, I can't remember when it was, whether it had happened by 2008. I believe it had. I think it was earlier than that mammalian transformation was achieved. Look at the picture of the little mice there. That's a litter of mice where one or the other parent had been transformed, and you've got some that clearly express the green fluorescent protein and some that don't. That's not photoshopped. Those really are looking as nice and beautiful and bright green as they can be. The thing that was difficult was getting plants to express fluorescent proteins, and I was very fortunate to work in the lab at Cambridge University where the guy figured out how to do it. When you would try and transform plant cells with the vector used to transform animal cells like this, it just wouldn't work. You'd analyze the plants later on and there'd be no fluorescence. And the guy that I worked for at Cambridge, his name is Jim Hasselhoff, figured out that there was a codon in the mammalian vector that plant cells recognized and cut, like they were removing a piece of the DNA, and it meant that the protein was never produced. So he fooled around with the sequence of the gene and finally got it working in plants. And that's been my research and the research of many, many other people over the last 20 years. Got some movies here for you, if I can get them to play. Bear with me. No, I didn't want to do that. I need my arrow pointer to make the movies play. This is a Drosophila embryo. For you people taking developmental biology or that just took developmental biology, you'll recognize the Drosophila embryo. Each green dot that you see is a nucleus of a cell, and what's going on here is that we're able to track the thousands and thousands of different cells moving around as the embryo goes through its early stages of development. This is the slime mold, Dictyostelium here. I'm not sure if it's marked with 2 colors. I think not. I think these cells here are marked with GFP, and the red is just an autofluorescence or a counterstain that's been used. Dictyostelium is something that makes evolutionary biologists scratch their heads, because for a lot of the time the cells live as individual protozoan, free-living single cells. But they're single-celled organisms that congregate when it's mating time, and they form a stalk and fruiting body, so-called. So it's a normally single-celled organism that comes together as a colonial organism when it's time to reproduce. The question evolutionary biologists ask is, why do some of the cells go into forming the stalk? Therefore, their genes aren't being passed to the next generation. It's something that would have had Charles Darwin scratching his head had he observed it. But watch this. There's the fruiting body forming at the tip of the stalk and sticking upright. Finally, the spores form up in here and are released. So absolutely fabulous. That's dictyostelium having been transformed with green fluorescent protein. So how do we do it? How do we get fluorescent protein into a plant or an animal? And I'll tell you, I was at Cornell University working away as a postdoctoral researcher in 1998, and people from another lab in the next building over were showing movies that they'd made at a conference, and they showed pictures of cells expressing green fluorescent protein. And I went running to them. I said, "You've got to give me some of that. Can I have a little tube of that stain?" And the woman looked at me and she said, "Idiot, it's not a stain. You have to use molecular biology, molecular genetics. You have to put it into the organism before the protein is expressed." I was very depressed about that at the time because I didn't know how to do molecular biology. Since then, and for that reason, I've taught myself to do molecular biology and spent a good deal of time learning how to clone just so that I could put fluorescent protein into different types of cells. So we're going to talk here about genetic modification of an organism for fluorescent protein expression. If you look at the picture, the yellow string that you see running along is a chromosome. And you guys know that along a chromosome, you find genes. So there's one of the genes of the organism. There's another one down there. Each gene has its own promoter region, which are indicated in blue. All you need to do, if you want to get a transgene into an organism, is cut the chromosome open, and that's what's represented by these arrows here, and insert the little bit of DNA that you've created. You remember I showed you the vector before, how you order the GFP. If you take the GFP sequence from that vector and put it into the chromosome here, along with a promoter that works in that organism, that means once the promoter is activated, that the GFP is produced. And that's the GFP. Here GFP fills the cell cytoplasm and the nucleoplasm because it contains no targeting information. But this is how all of the original experiments with fluorescent proteins were done. You made the protein went out into the cytoplasm, and it filled the cell cytoplasm so that when you would look with a fluorescence microscope down at a GFP containing cell, you would see a great big bright green blob. And the fascination with that sort of very quickly wore off. But it was important because it showed us that although GFP fills the nucleoplasm, it wasn't toxic to the cells. They could continue going through mitosis, as I explained earlier. So that's great. Now we can make cells that have GFP floating around in their cytoplasm. But we've got to go one step further than that if we want to make this a really useful tool in cell biology. So here's an example I prepared for you, and you'll notice the picture looks the same as the last picture. Here are the cut sites where we're inserting the fluorescent protein gene. There's the fluorescent protein gene and the promoter, just like in the last picture, but we have an intervening sequence of DNA that we've put in here. And I've labeled it known gene tissue specific. The example I'm using, the fluorescent protein gene can be fused to a known gene for subcellular targeting. Here, the known gene inserts into the nuclear envelope so that the envelope appears green. This white line running down the slide here is the nuclear envelope surrounding the nucleus. And I guess this is inside the nucleus because that's where the chromosome is. The known gene is a gene that makes a protein that we already know inserts into the nuclear envelope. So when you activate this promoter, this whole thing is transcribed, but it's transcribed in such a way that the known gene and the fluorescent protein stick together. There's no break in there. There's no stop codon at the end of this gene sequence. So you've got a start codon here, and you put a stop codon way down here. And look what happens. The known gene and the fluorescent protein remain stuck together like this. The known gene is the red one that goes to the nuclear envelope. It makes its way there naturally, and it's got this little green fluorescent protein hanging off of it like a tether. Right away, when you look down through the confocal microscope, you see the nuclear envelope lit up, in this case, nice and bright green. You get it? I hope you get that. I hope I've explained that well enough, because that is the basis of all of the research that we do. Here's a real-life example of a known gene for transforming living cells. And by the way, back when I was showing you the people that discovered GFP and won the Nobel Prize, I mentioned Thule Hazelrig, and I said she had done something important. She was amongst the first that figured out how to target GFP to a specific cellular organelle. So that's what I meant at the time. She's the one that figured out how to put a known gene sequence in here to fuse the GFP2. In this case, you're doing research and you want to make the Golgi apparatus green and fluorescent. What you need to do is find a gene that you know marks the membrane of the Golgi apparatus. Well, we have a candidate. There's a gene that's called sialyl-transferase, which goes right in here to the membrane of the Golgi apparatus. Sialyl-transferase is quite a big protein, but you know what? It's only the transmembrane domain of sialyl-transferase that goes and sticks into the membrane of the Golgi apparatus. The rest of it would just hang out in space. So people reasoned, and the people that did this reasoning, by the way, were at Oxford Brookes University. This is Professor Chris Hawes, who was my predecessor. He's the guy that taught this module before I took over, and he's the fellow that I came to work for in 2003. And that was a great honor for me to come to the place where they had figured out this trick. Take the gene sequence that codes for the transmembrane domain of the protein, put it in here. So this known gene doesn't have to be the entire gene sequence for a known gene. You use just the transmembrane domain. That's what the TMD stands for. Transmembrane domain fused to GFP makes a protein fusion that looks like this. You've got a GFP, which would normally just fill the cytoplasm, and it's got this bit that should go to the Golgi apparatus. Well, all you can do when you're doing these kind of experiments is transform the cell and see what happens. And lo and behold, if you put this genetic construct into the cell, and go look at the Golgi apparatus a couple of days later, the Golgi bodies are bright and green. And you think back, in the first part of the lecture, I showed you the movie of the little green Golgi bodies moving around inside of a plant cell. That was how it was done with the sialyl-transferase transmembrane domain. We call this, by the way, STGFP. And you'll find lots and lots of published research since where people mark the Golgi with STGFP. So, one last thing I want to say about this slide before moving on. This known gene that you're putting in here could be a whole gene, but it doesn't have to be. It can just be the part of the gene that conveys the subcellular targeting information. At the most rudimentary, we can target the endoplasmic reticulum just by putting 4 extra amino acids onto GFP. Then the GFP stays in the ER. At other times, and one of the big things in genomics research in the last 10 years or so has been, well, we still don't know what like 50-60% of the proteins in a given organism do. One thing you can do to characterize a protein is you can fuse the protein to GFP and you can see where in the cell the protein goes. And I've done this. We did this in a paper one time. We cloned 20 different genes, just took the whole gene sequence, cloned them so that GFP was attached to them, transformed the cells, and looked at where they went in the cells. And it was amazing. Some went to the endoplasmic reticulum, some to the Golgi, some to the plasma membrane, some to the tonoplast, which is the vacuole membrane. It was like absolutely amazing research. Really good to do. So that prompted me to make tables like this here. And it says here, I was very proud of this slide when it was made many years ago. What this slide shows is a subcellular compartment like endoplasmic reticulum. It shows the fusion protein that we use. I told you about the 4 amino acids that you can put on the end of GFP to light up the endoplasmic reticulum. HDEL are the amino acid codes for those 4. So take GFP, add HDEL to it, and you highlight the ER. Similarly, and you know from studying cell biology that chromatin is made up of DNA, of course, but also histone proteins. So you know that histones are a type of protein that go into the nucleus. If you take a histone and fuse it with a fluorescent protein, you can make the nucleus fluorescent. If you want to light up the plasma membrane of a cell, find a protein that you know goes to the plasma membrane. PIP stands for plasma membrane integral protein. It turns out to be a water transporter, and you can fuse it to whatever different color you want. And at this point we had done it to 3 different colors to light up the cell membrane. Another protein in cyan to light up the cell wall. Another protein that fuses to the cytoskeleton in different colors. Nowadays we have literally thousands of these different constructs. There are organisms like trypanosomes where people have fused every single gene with GFP just to see where in the cell the protein goes. So subcellular markers, and here's some of my own work here. I took this picture once upon a time in the very early days of this kind of work. That lab at Cambridge, somebody succeeded in getting the marking of the nuclei working, and I made what has become a pretty famous, if I do say so myself, picture of a root of a plant. The red stain is not red fluorescent protein, it's just a counter stain. It's a fluorochrome called propydium iodide. So I put the root of the plant in propydium iodide to stain the outside of the cells red, and we've got yellow fluorescent protein in this case expressed in every cell nucleus. Cell nucleus, histone protein 2b is fused to yellow fluorescent protein, so the chromatin in every one of these nuclei is marked. You can see the concentration of nuclei down around the lateral root tip. And if you remember back from first year when we looked at root tips, that's where the cell division zone is. That's where the cells are very small and compact, and you can really see that. You can see the nuclei way out here in the root hair, and in fact, that's what this was here. I was amazed. I made this movie and saw the nucleus actually moving around in these long, thin cells. You don't think of the nucleus as something that migrates around and changes shape like this. You think of the nucleus as a circle that sits in the middle of the cell. But look at that. Absolutely incredible. I've been meaning for years to go back and do some research on these big lobes of the nucleus that come out. It's a very deformable structure. And here we have the plasma membrane marker I was talking about that makes absolutely beautiful, bright, clear markings of the membrane at the outside of a cell. Well, that's the cell wall of plants marked with cyan fluorescent protein. Oh, that's the endoplasmic reticulum of the same cells marked with green fluorescent protein. Oh, look, that's the nuclei of the same cells marked with yellow fluorescent protein. I'd like to brag and say that the chloroplasts in those cells are marked with red fluorescent protein. They're not. But chloroplasts are autofluorescent. They naturally emit fluorescence in the red wavelength range. So I put it in there to make a 4-color image. Now, remember, there's something critical. Looking down at this little bit of plant stem, I've got all these different organelles marked. I've got them all marked at the same time. The plant's alive. We can look at this with a microscope. We can watch the endoplasmic reticulum. We can watch the nucleus, and we can observe what goes on in them as the plants function, as the plants do the business of living. So that was one of the key things about fluorescent proteins. They mark living cells, and you can use them in different color combinations. Different color combinations. Here are some examples from animal cells for you guys. HeLa cells with the, it might be endoplasmic reticulum, might be mitochondria, I can't tell. Nucleus marked in yellow. Nucleus is blue. Looks like mitochondria in red. Actin cytoskeleton in green, probably. Here we have, that could be endoplasmic reticulum and some other structure that might be the nucleus. All sorts of combinations. I'm sorry, I didn't even read to find out what these images are. These are just a few examples of the way that fluorescent proteins are being used to show these amazing combinations. Look at this one here. This blows me away. This is called brainbow for obvious reasons. It's a brain. It's probably the hippocampus of a brain. They've cut a thick section through a brain, so the animal in this case has been killed, but the brain cells are all still alive in the thick section. And they've done a trick. Now, I've told you that there's lots of different fluorescent proteins, but there's no way that you could specifically target one cell with blue fluorescent protein and another one with green fluorescent protein, another one with red. These guys have done an advanced trick. What they do is they have every single one of these hippocampal neurons expressing all of the same fluorescent proteins, but they've used clever molecular biology so that the level of the protein in each case that's made varies. So the one neuron might have a high level of cyan, a medium level of green, and a low level of red, whereas the neuron next door to it might have high red, high green, low cyan, if you see what I mean. And those ratios are generated randomly by the molecular biology trick that I'm not going to go into. So the random generation means that each neuron looks different from its neighbor when imaged, and these people have been able to go on and trace the course of hundreds of neurons at the same time. So this is really helping us learn how neurons map onto other neurons in the brain. Fascinating research. More movies. Because before I was showing you movies of whole organisms that were transformed with fluorescent proteins, but we can use fluorescent proteins to study subcellular processes. And just the couple of examples that I'm going to give you here are studying aspects of cell division. In the first movie, cyclin B1 expression during mitoses. Cyclin B1 fused to green fluorescent protein comes up as green. It's a checkpoint protein. It has to appear before the cells can go into prophase proper. So if you remember your cell cycle, you had GAP1, DNA synthesis, S phase, GAP2. There was a checkpoint before the cells can go into mitosis proper. Cyclin B1 is one of the checkpoint proteins, and it comes up, it's nice and bright green just before the cells start dividing, and as the cells are going through mitosis, as indicated by the red chromosomes that you'll see, watch the green level go down. See if you can spot all of that here. Cell with all the green starts going, there's metaphase, there's anaphase and telophase, and you'll notice that the green just wasn't there. The cyclin B1 goes away as the cell goes through cell division. I think it's amazing. And you can see that the next cell, the one over here, the level of cyclin B1 is still coming up as that cell gets ready to go into cell division. This one here I find really cute. I find it very balletic. I wish I could set it to music. Here are centromeres. So the centromere is the little center of the chromosome where the sister chromatids are held together until they separate at anaphase. So the centromeres are marked with green fluorescent protein. The picture's taken in black and white, but the fluorescence is green fluorescent protein. At metaphase, well, metaphase lasts for a long time. The chromosomes on the metaphase plate all line up and they dance around. And I think that's as the cell checks the integrity of the spindle apparatus to make sure that everything's connected up properly before anaphase happens. You want your chromosomes to separate so that they go to opposite poles of the cell. Let's just watch the chromosomes, the metaphase chromosomes, dancing around here. They're coming up to the metaphase plate. They're at mid-metaphase now, and they go into anaphase now. Ah, I love it. Let's watch that one more time, shall we? Okay. Oh, there it is. Can't get the movie back. Come into metaphase. You can even see that the centromeres are pairs, and then they separate. Absolutely beautiful. And finally, we want to study organelle dynamics, and this has been a lot of what people at Oxford Brookes have been doing for the last 20 years. Professor Chris Hawes that I told you about made the latter part of his career all about green fluorescent protein. He really popularized the idea of looking at things in what we call the secretory pathway, moving around and interacting. So I told you that you can mark the endoplasmic reticulum with green fluorescent protein just by adding 4 amino acids to the GFP sequence. And by the way, much later, in the semester, I'll be telling you all about exactly which 4 amino acids those are and why that works. This is a time-lapse movie that takes in 11 minutes in the life of a cell. I made this, and I was amazed when I watched it back. Absolutely beautiful. We've learned a lot about the endoplasmic reticulum structure and the dynamics of the different tubules within the ER just by watching movies like this over the years. This movie here is the same sort of thing. Lower magnification. The little red dots are the Golgi bodies running around inside of a plant cell. Absolutely fascinating to get a whole room full of scientists that are going, oh, look at this one here, or look at the way these two are associating. We've learned an awful lot about cells by looking at movies like this. We appreciate the beauty of these images that are being generated using fluorescent proteins and confocal microscopy, but we're doing real cutting-edge science, learning how proteins move around inside cells and how the different structures function with each other. And so I think, anyway, that the Nobel Prize in 2008 to those people I told you about was very, very well-deserved indeed, and I'm glad that they created fluorescent proteins for us to use. Okay, that's the end of the week 1 advanced imaging lecture. I'm going to come back next week and I'm going to talk about experimental techniques in bioimaging, and I'll do a bit at the end about electron microscopy as well. Okay, bye for now.